Hello, my name is Gabriel Gasque. I'm head of outreach at protocols.io. And today we will be discussing with Dr. Javier Monzo his recent PLOS ONE Lab Protocol article entitled "A Novel Laboratory Method to Stimulate Climatic Stress with Successful Application to Experiments with Medically Relevant Ticks," and the associated protocol in protocols.io. Dr. Monzo is an animal ecologist who was born in Colombia but grew up in New York City. He earned a bachelor's degree in biology from the City University of New York Queens College, where he conducted research on the singing behavior of canaries. He then earned a PhD in ecology and evolution for Stony Brook University, where he conducted research on genetics of coyote wolf hybrids. He began his studies on ticks as a postdoc in the Center for Infectious Diseases at Stony Brook University. Javier is presently a biology professor at Pepperdine University in Malibu, California, where he continues to conduct research on both coyotes and ticks. Javier, thank you so much for being with us.
Thank you so much for the invitation.
So to kick off, can you briefly explain what this protocol is about, how to use it, and why it is relevant?
Sure. We developed a novel, safe, stable, and convenient and economical method to isolate individual ticks and manipulate their microclimate. We use this protocol to investigate how climatic stress affects the survival, the behavior, and the physiology of ticks. And the protocol is relevant because ticks are medically relevant. Ticks are second to mosquitoes as the most prevalent vector of zoonotic disease-causing pathogens of humans.
So to understand better the significance, can you help us understand what was the state of the art when you decided to develop this protocol? Were there any other protocols similar to this one published? And if so, why these protocols didn't work for you?
Sure, there are previous protocols published that go back 60 to 70 years that manipulate the relative humidity specifically for studies of ticks or other arthropods. And it's not that previous published protocols didn't work. They do work, but they are inconvenient and potentially hazardous. Researchers typically use saturated or calibrated salt solutions to create a specific relative humidity environment. And this method requires a stock of laboratory-grade chemicals such as ammonium sulfate or calcium nitrate. And some of these salts and their solutions are corrosive and potentially hazardous, especially if they are swallowed or inhaled, spilled, or discarded improperly.
I see. And you spent this time developing and optimizing this protocol, but then you took a step forward and decided to make it public in protocols.io, and then have it peer-reviewed as a methodological paper in PLOS ONE. Can you tell us about your motivations to share, first of all, the protocol, and then to have it peer-reviewed?
Sure. Well, initially, we planned this large experiment with 3 species of ticks, 3 different temperatures, and 3 different humidities. And we planned to do it in 3 phases. So we began phase 1 in the spring of 2020. Well, while we were in the middle of phase 1, a little something called COVID happened, and all my students were sent home, and they were not allowed to come into the lab. So I had to finish phase 1 of the experiment by myself. Unfortunately, COVID lasted longer than anyone had expected, and students were not allowed on the campus of Pepperdine that summer, or that fall, or even the next spring. So finally, in the summer of 2021, we were allowed to have students back in our laboratories. and my student Antonio wanted to pick up where we left off with the tick project. But we introduced some changes, not to the physical setup of the protocol, but to the way that we evaluate tick survivorship. So after we completed phases 2 and 3, my colleague Lee Katz advised me to keep the phase 1 dataset separate from the phases 2 and 3 dataset because of the difference in how we evaluated survivorship. That's when I decided to publish the phase 1 data as a proof of concept lab protocol, and later the phases 2 and 3 data as a separate article. I saw that PLOS ONE partners with Protocols.io to publish novel lab protocols, so that's where I decided to submit our work.
And sorry, can you tell us about the feedback that you received from the peer reviewers, and if and how that feedback shaped the final version of your protocol?
Sure. Well, interestingly, the manuscript was first rejected by the PLOS ONE academic editor, and I appealed the decision with the PLOS ONE editor-in-chief and requested a different editor to handle the peer review. And in the second round of peer review, the main feedback was that we had not formally compared our protocol with the classical method of using the saturated salt, a method that had been used for 60 to 70 years. So this led us to conduct a new validation experiment to compare our protocol against the classical method side by side. And this yielded new evidence demonstrating that our protocol is substantially less costly and requires minimal preparation time compared to the classical method of using the saturated salt solution. And additionally, this side-by-side comparison showed that our method is faster at achieving equilibrium relative humidity. It's more accurate at achieving the target relative humidity. It's more resilient in returning to equilibrium after being disturbed, and it's more stable in both constant and in cycling temperatures. So our method represents an improvement over the classical method of using salt solutions, and that's what they wanted to see. They wanted to see that it's an improvement over what others had been using for decades.
So it seems then that the feedback that you received helped you set the bar and calibrate your protocol in relationship to published literature, and showed actually that, as you mentioned, it is an improvement.
Sure, because at first we just had this protocol, and so we submitted that. Here's our protocol, but they wanted to see not just that it works, but that it's an improvement over the classical method.
And changing now gears a little bit and focusing more on the protocol itself. While you were developing this protocol, was there anything that you tried that didn't work and didn't make it to the final version of the protocol? What variables did you try that weren't successful?
Sure. We tried a few things. We tried various ways to contain the ticks in their tubes by capping the tubes with something that permits airflow. So first we tried fastening a piece of mesh with a rubber band, and we also tried little mesh discs. And then finally, we just decided to get tubes with plastic caps that we drilled holes into the caps. And I have a few here to show you if you want to see.
Yeah, go ahead.
So basically, this is the setup, and let me back up. Here's the setup. This is basically just an airproof container, and we have these tubes, and inside of the tube is just a wooden dowel. And if you can see, here's a tick right there. It's probably crawling around. So we wanted to make sure that the caps have airflow, and yeah, that's where we tried all kinds of different things. And then my student just said, "Hey, why don't we just drill holes into them?"
And those are the same tubes that are depicted in the protocol.
These are the two.
Yeah. And for the user, what are the most critical steps in which they could potentially make mistakes?
Okay. Well, I think the most critical step is getting the ticks in their individual tubes, because ticks can be escape artists, and it's important to account for every single tick and not lose any. And this is especially important because ticks are potential carriers of disease, and the ticks we work on are not native to California. So we don't want to introduce any new ticks to California.
And are these ticks lab raised, or you get them shipped from, I don't know, the Northeast or from Europe?
All right, great question. So these ticks right here, these are lab raised. So we purchased them from the tick rearing facility of the Oklahoma State University. Uh, the ticks that we experimented on and published the protocol, they were lab reared. Uh, just this summer, we went to Oklahoma and New York to collect ticks from the wild, and we are repeating one of the experiments with wild caught ticks to see if their behavior and physiology is similar to the colony tick.
And are the ticks raised in the lab carriers of disease, or are the pathogens transmitted vertically or exclusively horizontally?
Well, some pathogens are transmitted vertically and some are transmitted horizontally. The ticks that are raised in the lab, the colony manager tells me that they are almost guaranteed to be pathogen free, right? But we should treat them as if they're not.
Okay.
So the animals that they're raised on, right? They're rabbits and sheep. They are known to be not carrying any tick-borne pathogen. But when I'm handling the ticks and when my students are handling the ticks, we treat all ticks as if they are carriers of disease. So we must account for every single tick.
I see. Yeah, I did my postdoc in a lab that worked with mosquitoes. And even though mosquitoes were lab raised and in principle pathogen free, we should handle them as if they weren't.
Exactly.
And
So I was just going to say an example of a horizontally transferred disease would be Lyme disease, where the bacterium Borrelia burgdorferi has to be picked up from another host. But another disease like Rocky Mountain spotted fever is caused by another bacterium, Rickettsia, which is transmitted through the ovaries from mother to offspring.
Thank you for the clarification. And since the publication of the protocol, have you made any further improvements? Have you had to troubleshoot it at any point?
Well, the troubleshooting, as I mentioned before, came early on with the caps, and the reason why that was important is because we need to evaluate the survivorship of the ticks as we stress them, and sometimes that is really obvious. Like here, all the ticks are obviously alive and moving around. Sometimes they look like they're dead, and we need to remove the tick, blow on it to see if it revives. And if it does revive, then we put it back into the experiment. And so that's why it was important for us to come up with a safe, easy, and fast way to uncap it and cap it back on. Because we were doing this with a lot of but since we published the protocol, not really. I mean, we've continued to use the protocol to complete phases 2 and 3 of the original experiment that I planned, and then we published the results of that experiment in the journal Ecosphere. And both the PLOS ONE and the Ecosphere papers were done with colony reared ticks. So now we are doing similar experiments with wild-caught ticks using the same protocol.
And will those wild-caught ticks be the same species as you tried in the paper that you reported already?
One of them is. So in the paper that we published in PLOS ONE and in Ecosphere, we used 3 species: the Lone Star tick, the American dog tick, and the black-legged tick. The experiment that we're doing now is with the Lone Star tick.
I see.
We
And
we
you
were able
expect,
to get those. We were able to get many of them in 2 weeks. 1 week in Oklahoma and 1 week in New York. We got so many of them and very few of the others, so that we're just repeating the experiment with the Lone Star tick.
and you expect your protocol to be transferable to any tick, regardless of whether it's caught in the wild or lab raised, and also independently of the species.
Sure. So we've already done the experiment. We're just analyzing the data on the wild caught tick, right? I mean, they're the same, except that we just got them from the wild. The only thing that we haven't tried yet is to do the experiment with other life stages like nymphs or even larvae. So far, we've only done the experiments on adult ticks. If we wanted to try the experiments with other life stages, I think we might need to troubleshoot a way to make sure that they don't escape, since they are tiny.
I see. And do they all feed from blood at all the stages?
Yeah, all 3 stages feed on blood. So the larvae, the nymphs, and the adults.
And they could all potentially transmit diseases.
Yes, the larvae can transmit only diseases that they inherited from their mothers.
Okay, I get it. And now, I mean, thank you so much for all the details about the ticks and about the protocol itself, and how do you expect it to translate, and how people, if they decide to implement it in other stages of the life cycle, they might need to optimize. But now, changing gears again, I would like you to mention what was the impact for you and your junior colleagues to get this protocol peer-reviewed as a standalone publication and published, obviously.
Sure, that's a great question. The protocol that we developed led to the PLOS ONE publication and the protocols IO protocol also being published. Later on, we continued to use it, and that led to another publication in Ecosphere. And both of those publications have a bunch of undergraduate authors, which is important because at Pepperdine, we only have an undergraduate program in biology. So all the research that we do is in collaboration with undergraduate students. And so the protocol has been really fruitful, not just for my career, but uh, to help kind of catapult the budding careers of other young scientists.
It's great to hear. And before we wrap up, is there anything that you would like to mention that we haven't discussed so far?
I thought that it would be important to mention that this whole idea was the idea of students. And I'm more of a behavioral ecologist that does work in the field, and I also do population genetics. And it was my student Will who came up with these ideas of manipulating the temperature and the relative humidity to stress ticks to see how they respond behaviorally and physiologically. And so we started playing around with various ways to do that, and we started playing around with the saw because that's how it's done. That's how it's been done for decades. And that's when we saw that this is really messy. It's difficult. It's um the saws can be difficult to handle. They need special care in handling them. Um, anyway, so Will laid the groundwork. My student Will, who is a co-author of the PLOS ONE paper, he laid the groundwork for these ideas. But then he graduated. I then got caught up in studies of coyotes for a few years, and then another group of students wanted to revive these ideas for a class project. So I suppose that they were first motivated to optimize the protocol so they can get a good grade. and they did. But the interesting thing is that even after the course was over and they got their grade, they wanted to continue optimizing the protocol and continuing the research. And so that's what led to that PLOS ONE article. And those students are the other co-authors of the PLOS ONE article.
Well, there was a lot of drive and motivation, and I'm really happy to see that it paid off.
Yeah, yeah. I was really glad. It was a very fruitful effort, and I'm glad that it was primarily driven by the students' curiosity and ideas.
Well, Javier, it's been very enlightening. I appreciate your time and your candidness. And thank you so much.
Thank you, Gabriel. I really enjoyed this conversation.